Good morning, students. Hope you are doing well. Today we shall discuss one important topic from molecular basis of inheritance. The topic name is Lac Operon concept, and this Lac Operon concept we are going to understand this concept in terms of three categories like introduction, structural aspects, and mechanism. And when it comes to the introduction, all of us very familiar about one concept, that is central dogma of the cell. In the central dogma of the cell, DNA has ability to replicate itself, and the DNA undergoes transcription to produce mRNA, and mRNA undergoes translation to produce protein. So this is called the central dogma of the cell. The ultimate aim of the central dogma of the cell is to express gene in the form of protein. The gene is a form of gene as having a special character, and this character is manifested in the form of protein. So here my point is the DNA contains hundreds and thousands of the genes, and all genes are present in the DNA right from the childhood to the adult age, and the cell contains all these genes every time, every single minute of any living organism's life. But not all genes are expressing or manifesting its concerned protein. Only few genes are expressing in the form of protein at particular time. That means each gene is having a specific timing for its expression in the form of protein. And how the timing is happening, and who are all the factors who are controlling or who are regulating these genes to express in the form of protein in their specified timing. And these things these points are going to clarified if we understand the process of gene regulation by taking Lac Operon as an example. The gene regulation, we can understand the gene regulation very clearly by understanding Lac Operon concept. And this Lac Operon is related to the prokaryotic bacteria, that is E. coli bacteria. So in the E. coli bacteria, this Lac Operon is present, and that is about introductory part. And the idea, the objective of the introductory part is, so there are many genes expressing in different timings. How this regulation is possible, and this we are going to understand from step 2 and step 3 of this topic. In step 2, that is structural aspects, before getting to the third point, there comes the structural aspects. In the structural aspects, see, this is the DNA sequence of E. coli, and this DNA sequence contains two important zones like regulatory sequence and the structural gene. And this regulatory sequence contains promoter, inhibitor, again promoter, and the operator, and each one is having specific function. The promoter is the prime place where RNA lands and goes for the process of the transcription, whereas I refers to the inhibitor, and this inhibitor, the prime function of the inhibitor undergoes this inhibitor gene undergoes always turn on and undergoes transcription, produce an mRNA, and this mRNA undergoes translation and produces a concerned protein, that is repressor protein. Because this inhibitor gene always turn on, this is called constitutive gene, and these constitutive genes are the genes which are always under functional mode. So based on this, we can call it as constitutive gene. So this inhibitor gene is a constitutive gene, and the promoter is the place where RNA polymerase attaches and helps in the process of the transcription. Operator zone is also a kind of DNA sequence, and this operator sequence or DNA sequence acts as a switch for the process of transcription of this structural gene. So this operator zone is a kind of DNA sequence, helps as or functions as a switch, and that switch guides whether transcription should happen or not, right? So likewise, each sequence, like promoter sequence has its own purpose, operator sequence has its own purpose, and inhibitor gene is having its own purpose to produce repressor protein. And this repressor protein plays a vital role, a pivotal role in regulating entire system of this structural gene. The structural genes contains 3 genes, and these genes supposed to manifest, transcribe and translate to produce its concerned protein, and that regulatory mechanism being done by the repressor protein. And this is key point, this is key gene, key protein that plays major role in the process of regulating ZYA genes, right? This is a regulatory sequence when it comes to the structural gene, and this structural gene is a polycistronic type. Polycistronic type of gene meaning having more than one gene with one having present in one mRNA sequence, or one gene, more than one gene presenting in one mRNA sequence. It's a polycistronic type of structural gene, whereas this one regulatory inhibitor gene is a monocistronic type, whereas this one structural gene is a polycistronic type, contains ZYA gene, and here Z refers to beta galactosidase. It is beta galactosidase, and Y refers to permease, and A refers to transacetylase. So that means what? So gene Z produces, translates a beta galactosidase enzyme, and gene Y translates permease enzyme, and gene A translates transacetylase enzyme. So this is a structural gene, and this is a regulatory gene. This structural gene is called as facultative gene or non-constitutive gene, meaning maybe it is on or off. We cannot say that always structural gene is on, turned on, or we cannot say that always structural gene turns off. No, based on the situation, conditions, based on the need of the cell, this structural gene may be present, turns on or turns off. Based on this, we can call it as facultative gene or non-constitutive gene, whereas repressor protein, that inhibitor gene, is called as constitutive gene. So this is the structural aspects of the DNA sequence in the Lac Operon concept. Now we'll see third important aspect, that is mechanism. And this mechanism we can understand this mechanism in 2 cases, case 1 and case 2. Case 1 is when this operon turns on, and let's take case 1. In case 1, this is mechanism. Now it is mechanism, it is case 1. Case 1, same operon, like promoter, inhibitor, promoter and operator, Z, Y and A. And in case 1, so that means what? In the case 1, it is in the absence of inducer. In the absence of inducer. So here comes one different word, that is inducer. So we have seen inhibitor, we have seen structural genes, and there is one more word called inducer, that the very purpose of inducer is, it is to induce the process or mechanism of Lac Operon, that is transcription of ZYA genes. So the inducer here called as lactose. See, the very concept, the very title name of our concept is Lac Operon. The lac refers to the lactose, operon refers to a cluster of genes which are controlled by one regulatory sequence. So this is operon, and this is the meaning of the operon, whereas the lac refers to the lactose, and the lactose is acts as a inducer, and biochemically it is the it is of two more, it is biochemically this lactose contains two monosaccharides, like it is a disaccharide, and this disaccharide composed of one monosaccharide called glucose and galactose. Each one is one monosaccharide. Combinedly together we can call it as a disaccharide, and the lactose is a disaccharide which acts as an inducer. That means what? Whenever the bacteria feeds on lactose, whenever the bacteria feeds on the lactose, because since the lactose is it's a good nutritious food material for the lactose, and whenever it feeds on lactose, the lactose enters into the bacterial cell, and the lactose molecule which entered into the bacterial cell acts as inducer to trigger this Lac Operon method. That means to trigger the transcription of ZYA gene. So in the absence of the lactose, when the bacteria stops feeding on the lactose, there won't be any inducer called lactose. Since there is no inducer, the Lac Operon, the genes of ZYA genes remains silent. They won't manifest, they won't transcribe concerned mRNA or translate into respective enzymes of the proteins. Only in the presence of the lactose, that is in the presence of inducer, the triggers, these lactose triggers for the transcription of ZYA gene. So we are seeing the case where the inducer is absent, that means lactose is absent, that's bacteria feeding on some other molecule other than the lactose. When lactose is absent, that means inducer is absent. What happens here? As usual, the inhibitor gene is present here, and this inhibitor gene, irrespective of presence and absence of the lactose, inhibitor gene always transcribes into inhibitor mRNA, or you can call repressor mRNA, and this repressor mRNA translates into repressor protein. We are talking about the case where lactose is absent, that means inducer is absent. Since the inducer is absent, there is no possibility of entering lactose or inducer. So then and as usual, so as per the daily activity, this inducer, that inhibitor gene produces by translating an repressor protein, and this repressor protein has ability, this repressor protein which is produced during the translation process and goes and binds to the operator gene here, operator sequence there. Since the repressor protein binds to the operator sequence and the promoter sequence, the promoter sequence attached with the RNA polymerase here, and here comes the RNA polymerase comes and attached to the promoter during the process of the transcription. And since operator zone occupied with the repressor protein, as the process in the course of transcription, this RNA polymerase which is present in the promoter sequence here tries to move further, since it is blocked by, since operator zone blocked by repressor protein, RNA polymerase cannot make a move further, and that's how the process of the transcription stops here. So who is going to stop this process of transcription? The repressor protein. Since the repressor protein is stopping the process of the transcription, this repressor protein is very, very crucial in regulating the process of concerned genes. That means what? There is somebody who is taking the charge of regulation of each gene in a DNA. That is how all the genes that are present in the DNA being regulated by different regulators. If there were no regulators, every gene will transcribe and translate concerned protein, and that will lead to some abnormal events in a living system. For everything there is specific timing. For every protein there is specific timing. That means what? Let's take a plant as an example. There is a phase for the vegetative phase, there is a phase for the reproductive phase, and the reproductive phase is always followed by certain hormones, and these hormones are being released by concerned genes. And that particular stage where the plant decided to enter in the reproductive phase, in that particular phase only the concerned genes will express concerned protein in the form of hormones, and those hormones will only drag or takes this plant from vegetative phase to the reproductive phase. Likewise, some regulatory methods are there. Here the regulatory part is being taken care of by repressor protein. So this repressor protein is stopping the functioning of ZYA genes, right? So why? Because inducer is absent. That means what? The functioning of ZYA possible only when bacteria cell needs its concerned enzymes, because ZYA proteins are concerned with certain enzymes, and those enzymes are only function on lactose molecule. When lactose molecule, there is no point of having all the ZYA proteins inside the cell. Only when bacteria feeds on the lactose molecule, that is the time where concerned enzymes are required. So that is the reason this repressor protein stops the functioning of ZYA genes here. So this is case 1. Let's see the case 2. In case 2, it is case 1, it is absence of inducer, whereas case 2 so in the case 2 it is presence of inducer. So in the presence of inducer means what? Lactose is present. Here lactose is present. When bacteria feeds on the lactose enters into the bacteria. Here what happens? When lactose enters into the bacteria, this lactose converts into allolactose, and this is possible by the enzyme beta galactosidase. So this beta galactosidase is an enzyme which converts the lactose into allolactose, and this allolactose is a direct inducer for triggering the process of ZYA transcription. And lactose acts as an indirect inducer, whereas allolactose acts as direct inducer for the transcription of ZYA genes. What happens here? See here, so when lactose enters into the bacteria, so this, as part of the schedule, this I gene produces its mRNA, that is repressor mRNA, and this mRNA undergoes translation, produces repressor protein. So since after producing repressor protein, the lactose or allolactose molecule which enters into the bacterial cell. This allolactose molecule will comes and binds with the this repressor protein. So because this allolactose comes and binds with the repressor protein, as part of the schedule, this repressor protein along with the allolactose tries to right, yeah, so it tries to attach here. So at operator zone is trying to attach here at the operator zone. Since the repressor protein conformation got changed because of this allolactose molecule, this repressor protein would not fit here. So this repressor protein would not fit here, and there won't be any proper attachment of repressor protein at operator sequence, and because of this, yes, RNA polymerase which is already there, so RNA polymerase which is already there at the promoter sequence will try to move further for the process of the transcription. Thereby transcription takes place, and mRNA will form for the process of transcription. And this mRNA will contain ZYA genes, and this transcription, after transcription, this mRNA undergoes translation, produces beta galactosidase protein and permease and transacetylase. So after translation, from this mRNA we could get beta galactosidase enzyme, permease and transacetylase enzyme, because this is the time where bacteria needs these enzymes, because now these enzymes have specific function to act on. Prior to this, these enzymes necessity is very small or nullified. Bacteria doesn't need any use of these enzymes, only during feeding only the bacteria feeds on this lactose molecule. These enzymes are much required for the process of the digestion of this disaccharide molecule, and after acting these enzymes, this disaccharide molecule will broken down into monosaccharide molecule. So that is how the regulation is being done by the regulatory sequence. In the absence of the regulatory sequence, we cannot expect this proper functioning of these genes. Only because of the regulatory sequence, the genes are regulated, are scheduled to function in its own timing. Here the timing has come where the lactose induced the process of the transcription here. So the allolactose comes and binds to the repressor protein, and the repressor protein comes and tries to bind in the operator sequence, and because of the change in the conformation, the operator sequence will not allow this repressor protein, the modified repressor protein. Thereby the RNA polymerase will get good opportunity to further to move further for the process of transcription. As it transcribes mRNA, it undergoes translation, produces three different enzymes: beta galactosidase, permease and transacetylase. And beta galactosidase, an enzyme, helps in breaking of works and acts on lactose to break down into glucose and galactose. So this beta galactosidase breaks down the lactose into glucose and the galactose molecule, whereas permease helps in transfer of lactose molecule into the bacterial cell. So it permits lactose molecule into the bacterial cell, whereas transacetylase in enzyme that helps in transferring acetyl group from galactose molecule. So for transfer of acetyl group, transacetylase helps in transfer of acetyl group, and whereas permease helps in permitting the lactose into the bacteria, and beta galactosidase acts on lactose to break down into the glucose and the galactose molecule, and that's how all enzymes work together for the process of digestion of the lactose molecule. And this is called what do you call, because it is positive regulation. So because the regulation, the what do you call, the switch, what do you call, the regulatory sequence turned on, that means the regulatory sequence is ZYA, the structural gene turned on for its function, it is called positive regulation, whereas when the ZYA structural gene turned off, it is called negative regulation. So togetherly, by positive regulation and negative regulation, entire process of lactose digestion or metabolism controlled by this bacteria. So likewise, so this is the final mechanism of Lac Operon. So finally, in nutshell, the idea is the concept of this session is to understand how the regulation process is happening, and this is possible with the regulatory sequence along with the structural gene, the regulatory sequence regulating entire process of this ZYA genes, and in presence of the lactose, case 1, in the absence of the lactose is case 2, and combinedly by clubbing of these two mechanisms, complete digestion of the lactose takes place here. Thank you one and all.